Alright, so welcome back. We are still in Module 9. However, today we're going to be going over Part 2 of our week's topic here. So we're still doing taxonomy and systemics, and so we're going to be covering the next section of that we had for our required readings. So make sure you're doing your reading. You're on pages 147 to 165, all about taxonomy, systemics, cladograms, phylogenetic trees. You're doing all of that fun stuff this week. Okay, so just a few quick reminders before we continue on to the new stuff today. There's a few new assignments. Well, actually, this first one here is an older assignment, but I want to make sure that you guys get it all turned in and ready to go. So your presentation topics for your presentations that are going to be due in a week or so here, I want to make sure that you get your presentation topics selected. So that is due on April 10th. So make sure you're picking out a topic that you can talk 3 to 5 minutes about. We're going to do recordings if possible, so you'll be able to record yourself giving a presentation, kind of like either how I'm doing here or with your smartphone. And if that's not an option, you can go ahead and submit your PowerPoint presentation and give me your presentation over the phone. Or there's a few other options that you'll be able to do. We can make it work. So anyway, to select your topic, if you haven't already, go under Cobra in your content in Module 8, and there is a discussion thread that says PowerPoint topics, and I want you all to post your topic. First come, first serve on topic choices, so if there's something you really want to do, get on in there and put it down so that way nobody else can take your topic. We don't want any duplicates. The next thing on our list here is our module number 9 quiz. So that is going to be due Tuesday, April 14th. So 1 week from April 7th. And so there's 10 random questions out of a possible 29 or so. It's open book, 20 minutes long. This is going to be all the stuff that we cover this week about taxonomy and systemics. It's going to be cladograms, phylogenetic trees, all of the stuff that goes along with the study of taxonomy. And everything that we covered this week, week April 6th through the 10th. So that's the week that we're going to be focusing on here. So another thing on your list of what is due is your lab number 9, your communicules lab. So that is the cute little slug creatures that you have to do for lab this week. You're going to be cutting them out. Placing them on a phylogenetic tree. You have fossils and living communicules. I believe there's 2 or 3 parts for that lab. That's going to be due on Tuesday, April 14th. You guys are really innovative with taking photos of your phylogenetic trees and filling out your lab on the computer, so that's great. Just keep doing what works and we'll make it through. And then the last thing that I assigned this week is an evolving switches video. So this is a video, it's about 15 minutes long. You can access it by going to the website that I provided in your Cobra and in your email when I gave you your updated list of everything that's due. The link is there. It's a 15-minute video about stickleback fish, and so this evolving switches video, you're supposed to watch it, and then there is a 9-question quiz where you write in the answers from what you watched in the video. So make sure you're doing that because that's also easy points and it's really quick and easy to do. Okay, so again, your reading pages here, 147 to 165. Here's all of the sections that is included in your book. Today we're going to be covering pretty much only cladistics. So cladograms, how to create a cladogram, what a cladogram is, different components of that. And so that's what we're covering today. And then our last lecture will cover the last 2 sections, which is molecular clocks and horizontal gene transfer. Okay, so cladistics. So what is cladistics? So cladistics is the study and the classification of a species that we have here that's based on evolutionary relationships. So we're looking at relationships between a few different species, studying how that fits in, kind of like putting together a puzzle. Now, there is one thing to note when we're talking about a cladistic approach, when we're making a tree here, a cladogram. So they do discriminate among possible trees, because even though they consider various possible pathways of changes, they do tend to choose the tree that requires the least complex explanation. So the easiest tree to explain is the one that is typically the one that is selected, because in a lot of cases that is the one that is correct. When you talk about making cladograms and talking about evolution based on different species, typically you see the path of least resistance is the one that is likely to be correct. And so we're also going to talk about making phylogenetic trees or cladograms. They are a little bit different, but we'll go into the differences here, primarily because the cladogram is basing it upon evolutionary relationships, where phylogenetic trees looks at time that has passed over the course of how long it has taken for species to be developed. Okay, so again here, I have a video. You've seen one of this guy's videos before, but he did a really great job in creating a cladogram. Now, I don't have the software that he has to make all of this stuff look really nice here, but he does a really great job of creating a cladogram from scratch for a completely made-up species that he has. And so I'm going to play this cladogram here, this cladogram video here, and then hopefully you'll be able to understand how to generate one.
Hi, it's Mr. Anderson, and in this podcast I'm going to talk about the cladogram. Cladogram is essentially taking characteristics that are shared by organisms and then putting together a hypothesis of how you think they may be related, or a phylogenetic tree. It's not a phylogenetic tree, it's just a guess at how that might occur. And so luckily I was just working in some pond water yesterday, and I discovered these 7 creatures, and I've named them after myself, of course. They're called Andersenii aquaticus, and they're all subspecies of that. But basically they have a lot of different characteristics. And so if we were to put those together in a cladogram, well, when you were growing up, you probably watched Sesame Street, and they used to play a game called One of These Things is Not Like the Other. And so that's a good way to think about a cladogram. And so basically, which of these doesn't look like the others, which of these is an outgroup? Well, you should be able to figure this out. First one would be C, and then try to guess before I actually click on it. The next one that would branch off according to this would be B, because it doesn't have a I, but we're going to have that in G. So next would be G. Try to play along with me. What's going to be the next one? That's right, it's going to be D. And then after that, A. And then after that, F. And then finally after that, we have E. So you could probably do that. Just put them in order, looking at what characteristics they have. So the outgroup is going to be the one that doesn't have any of these characteristics, or maybe one. And so basically a cladogram is going to look like this. It's going to be a hypothesis of how these things came to be. And so I could look down here and I could say, well, right here there's one characteristic, and that would be that there is a cell membrane. In other words, all of them have a cell membrane. And so to show you how this is read, basically everything that branches out from this point is going to be a cell membrane. What is this branching point? Well, you can think of that as a common ancestor between all of the organisms that are on here. Or if I were to go right here, this would be a circle. Or if I were to move up a little bit, what's going to be the next thing that comes? The next thing is going to be the eye. And so I could keep going all the way up. These are characteristics that are added, and we use those to make a cladogram. Now, there's some interesting things you should know. So if we look at this cladogram and say that it's true, who's more related? Is F more related to E, or is it more related to A? That's a really good question. And so basically, how do we know which it's related to? And I would say the right answer is E. You have to understand that a cladogram, or a phylogenetic tree for that matter, time is going to go in this direction. So we're going to put time on the side. In other words, this is going to be the most ancient common ancestor, and then we're going to work progressively up. And so that means that F and E would share a common ancestor right here, but F and A are going to share a common ancestor all the way back down here. And since this is way farther back in time, we would say that F is more related to E. But this is a pretty simplistic one, and again, it's just a guess as to how it might occur. A tougher question might be, well, let's add a new one. Let's say I discover something like this. This is species H, or subspecies H. Where would you put this on this cladogram? And when I talk to students in class, there's 3 areas, or 4 areas where they might want to add that. So some people think we should add it kind of like branching off here. Some think we should do it right here, branching off D. Some think it should be something like this, and some think it should look something like that. And so these are all plausible. In other words, these are all guesses. And we could put way more in there than this. But what's the quote-unquote right answer? Well, if I had to put this somewhere, what I would do is I would put it right here. In other words, I would have H branching off here. And the reason why is that if I were to put it way back here, before the I evolved because we didn't have that I, well, it could have occurred like that, but it would be odd that it would also evolve the same exact teeth and the same defensive spikes that we find farther up. And so a more plausible guess would be that it branched off of this A and then it lost its I. Now, how could it lose its I? Well, if we were to put one of these in an area where there's no light, then the I wouldn't be an advantage. And so lots of times when we're adding a cladogram, we just add characteristics that are added, but know this, that it's just as likely to lose something that we've gained as to gain something new. And so this would be kind of a pretty good guess as to where H goes. And what I'm talking about now is Occam's razor. In other words, I'm talking about the theory of parsimony. What in the heck does that even mean? Well, it basically means this. If all things are equal, the simplest explanation for how that occurred is probably the most right. In other words, if I come out in the morning and there's a glass of milk that's spilled in the kitchen, I could come up with a bunch of different hypotheses. It could be aliens, it could be Bigfoot, it could be a massive earthquake, or it could be my cat. And if I were to look at all of these, well, it's most likely going to be my cat. Doesn't mean that it's right. It doesn't mean that aliens didn't come down and spill my milk. It just means that it's more than likely the cat that did it. And so when you're building a cladogram, you kind of try to do that. And you try to use this idea of parsimony, the idea that the simplest explanation is the best. So let me show you an example of one of these. This would be a cladogram that's created for primates. And if we look at this one again to go back, in general in biology we used to use all these characteristics to put organisms in a cladogram, physical characteristics, but you can imagine that's not a great way to classify life or to even show its evolutionary relationships. Today we now have DNA, we have genetics. And so you can create a cladogram like this by just sequencing the DNA in all these different organisms and then throwing them into a computer, and it can create a plausible kind of a cladogram. And so where are you? You're right here on the human side. And so if we were to go across from the left, so if we look at these two, the lemur and the loris, they share a common ancestor right here, but they also show a distant common ancestor with all of the primates on here all the way back here. If we were to work across to the tarsier or the New World and Old World monkeys is kind of a weird term. New World is a South American monkey. So an example could be the squirrel monkey. An Old World monkey would be like a baboon. So all of these things have tails, and now we move up to the apes. The first one would be the gibbon. That would be this first branch point. Then we have the orangutan, the gorilla, the chimp, and we could put bonobo branching off here, and then we finally have the humans. And so again, this is just a cladogram. It's a hypothesis of how these things are related, and you're looking at characteristics that are shared by all of these, but the big characteristics we're looking at now are homologous structures of DNA. In other words, where the genetics match up, that means they had a common ancestor. Does it mean that we turned from gorillas to humans? No, it just means that we had an ancestor way back at this point. And so those are cladograms, and I hope that's helpful.
So that was a great example of how to build a cladogram. So I thought he did a really great job of arranging it and kind of explaining about how ancestors, where they fit in, where their role is when you do build a cladogram, to note where there's a common ancestor and an ancestor that's more closely related or a derived ancestor. Okay, so he mentioned the homologous traits. So I talked about that in my last lecture, the homologous traits. We also considered those a character. So when I say a character, I am referring to a specific homologous trait or a trait that exists on an organism's body. And it may exist in two or more character states. So there's two ways in which those traits or those characters may exist. So the first one is a simple seomorphy. So this is basically a shared primitive character. So this is what the fancy name of it is, the simple seomorphy. So what this is a trait or a character that is shared by two or more taxa and inherited from ancestors that are older than their last common ancestor. And we'll look at what simple seomorphy looks like on a cladogram here in a second here. Okay, and so the second character state that exists is called a synapomorphy. So this is a shared derived character. So instead of a shared primitive character, which is the simple seomorphy, now we have a shared derived character. So what this is a character that is shared by two or more species or taxa that has been originated in the most recent common ancestor. So it came out of the most recent common ancestor. And the basis of this, when in terms of a cladogram or a cladistic approach, is to analyze these shared derived characters to kind of determine the pathway that gave rise to those species. So it's helping us to determine, how those species came to be. So on the next slide here, we're going to look at the simple seomorphy and a synapomorphy and how it looks on a cladogram. Okay, so here you can see we have two little small cladograms. We have the top one and the bottom one. So the top one is showing us a synapomorphy and the bottom one is showing us a simple seomorphy. So you can see here that the hollow circles are derived traits. We have the filled in blacked in circles. Those are our ancestral traits. And then you can't really see it too well here because it's not, we're not going to use that right now, but that's an evolutionary novelty, is that third hollow. It's actually filled in yellow, like a highlighted color, but you can't really see it, but it's not on this list. So we're just going to pretend like that one doesn't exist. So we're just going to look at the derived traits, which are the hollow circles, and the filled in black little circles, which are the ancestor traits. Okay? So let's look at the synapomorphy first, so the top cladogram. So you can see there are 3 derived traits, and then the rest are ancestral traits. So that synapomorphy here is those shared derived traits, if you remember. So that's showing us how these species here at the end of this cladogram at the tips of the trees came to be. So those 3 hollow circles have a specific derived character or derived trait. And that little section here of those 3 hollow circles is our synapomorphy. Okay, so for the second cladogram, we have a simple seomorphy. So we have the example here. We have one hollow circle, so that's our one derived trait at the very end of the tree tip here, and then all the other 6 circles are filled in blacked out circles. Those are all of our ancestral traits. And so you can see our simple seomorphy is encompassing those 3 filled in circles at the tip of our cladogram, which is basically showing us that these traits have come from an ancestor farther away down the evolutionary chain. So you can see all of the ancestors have those ancestral traits, and all of the species that were produced from those ancestors also have that ancestral trait. So you can see all 3 of those species have that. And so an example of this might be a common eye, like an eyeball. So all of those ancestors have eyes, and all of the descendants themselves also share this ancestral trait, and they all themselves have eyes as well. Okay. So here's an example You can see in this figure here, figure 26.8, it's showing you an example of a cladogram. So you can see species A here is this, it's like a rat looking thing. So species A has 2 eyes and 2 front legs. So those are traits that this ancestor has. Now if you look, you can see that from this ancestor species, species A, we get species B, C, D, E, F, and G. So from ancestor A, we have a split of 2 species. We have species B and species C. So species B looks like some sort of primitive dolphin, and species C looks like some sort of primitive mouse or rat. Very similar to what the ancestral species looks like. Now you can see in both of species B and species C, they both have 2 sets of eyes. So that would be considered our ancestral trait so far. So this is something that both species have. It's coming from the ancestor. Now you can see here in the leg section, this is where it's changing up a little bit. So species B, instead of having 2 front legs, it has 2 front flippers. And so species C still has its 2 front legs. So now we don't quite have the ancestral trait anymore in species B as we had coming from species A and species C. So those 2 still have the 2 front legs, but species B has now the 2 front flippers. So now you're getting into something that's going to be eventually a derived characteristic. Because if you look, after species B develops into 2 separate species, or diverges into 2 separate species, we have a whale and a dolphin, species D and species E. Those 2 species still have 2 eyes, just like the ancestor, so it's still an ancestral trait, but now they also have 2 flippers. So those 3 species, D, E, and B, all have flippers. So that is our derived character, our derived trait. Now it's different than the 2 front legs that we have in species C that came from species A, and after species C diverges into 2 separate species, you have species F and species G. So we have some sort of vole or something as species F, and some sort of mouse for species G. They both have 2 sets of eyes, just like their ancestors, so that's our ancestral trait. But they also have 2 front legs, so that is also our ancestral trait. So that is staying the same. They don't have the flippers, which is that derived trait. So hopefully that clarified it a little bit because we were able to break it down. But we'll talk more about it here in the next slides as well. So some things that you did see in the last slide, where are some of these terms here that I'm going to talk about. So we saw branch points. So this is the area in a cladogram where 2 species will differ in shared derived characters. So this is where you might see something branch off that's a little bit different when we're talking about those shared derived characteristics, like flippers or some other derived character. We also have an in-group. So this is a term that we use to discuss a group of species that we are interested in. So when we're talking about an in-group, typically they'll circle a group, or they'll mention which species that they're talking about in a particular question. So when we're talking about something, it's like an inclusive group. We also have the outgroup. So an outgroup is a species or a group of species that's assumed to have diverged before the species in the in-group. So it kind of branched off and separated before that in-group species that we're interested in, that we're talking about here. So that outgroup is something we wouldn't be discussing. It's determined that maybe they developed some characters, or they diverged before the group that we're talking about in an in-group, for instance. Typically, when we talk about an outgroup, it will lack 1 or more shared derived characters that are found within the in-group. So if we're talking about something specific, a specific trait, the outgroup typically won't have that trait. So In your quiz questions, they might have images there, and they'll have an in-group that they're referencing or circled, and then they won't be discussing the out-group because typically they won't have that shared derived character. Okay, so in this figure, figure 26.9, now we're using different morphological features or morphological traits in order to develop a cladogram. So you've seen something like this a little bit before. In this case here, we're using different features to arrange these 5 different species: the lancelet, the lamprey, the salmon, the lizard, and the rabbit. So we're using those 5 species, and we're using a few different traits. So we're using the notochord, the vertebrae, the hinged jaw, the tetrapod, and the mammary glands. So we're using those characters to kind of create this cladogram. So you can see in the chart above the cladogram, you can see the 5 species there and the 6 different traits that we have, or the 5 different traits that we have. And you can see which species has it and which species doesn't. And so if you just had a bunch of species and you knew certain features that they had, you could start to put them into columns. And then after that, after you determine who has what, then you can start to create a cladogram. So for example, the 5 species that we have: the lancelet, the lamprey, the salmon, the lizard, and the rabbit. So we can say, okay, which species has a notochord, which is basically like a cord that's running in your spinal column here. So we know that all 5 of these species here have a notochord, so they all have this feature. So we're going to say yes, they all have notochords. So the next trait, let's look at vertebrae. So vertebrae, obviously the bones in your backbone. We're going to say who has a vertebrae. So we know that only 4 species has a vertebrae. The lancelet does not have a vertebrae, but the lamprey, the salmon, the lizard, and the rabbit all do. So we'll put them in the yes column. Next, we're going to look at the next trait, the hinged jaw. Who has a hinged jaw of these 5 species? The lancelet and the lamprey do not have hinged jaws, but the other 3 species, the salmon, the lizard, and the rabbit, do in fact have hinged jaws. So we'll put them in the yes column here. Who is a tetrapod? So who has 4 limbs, walks on all 4s here? So we know that the salmon does not, but the lizard and the rabbit do. So we are going to put them in the yes column. And then lastly, which one of these species have mammary glands? Well, the lancelet, the lamprey, the salmon, and the lizard don't have mammary glands, but the rabbit does. And so now we know who has these traits and who does not have these traits. So we can now form a cladogram to help determine what it would look like if we were to put them all based on their evolutionary relationships in this chart. So we could see the lancelet was probably one of the first species to exist, and they had a notochord, but so does everybody else. So we put that notochord at the very bottom here of our cladogram. And then we move up the line. We have the vertebrae came next. We know that the lancelet did not have the vertebrae, but the lamprey, salmon, lizard, and rabbit all have the vertebrae. So then they're going to come after that vertebrae trait. Same thing with the hinged jaw. We know lamprey doesn't, so we'll put the hinged jaw trait after the lamprey already came into existence, so that way it shows us that the salmon, the lizard, and the rabbit all have hinged jaws. And then we have the tetrapod. So who's a tetrapod? With the lizard and the rabbit are. So we're going to put that trait after the salmon already exists in the cladogram, because that trait came after the salmon was already formed. So we're going to put that tetrapod there. And then lastly, the mammary glands. We know the rabbit's the only one that has the mammary glands. So we're going to put that trait right before where the rabbit exists, but after where the lizard exists, because the lizard does not have those mammary glands. So that's just an easy way to kind of place your traits in accordance with who was developed first and what their evolutionary relationships look like. So one other thing with cladograms is they can also be constructed with gene sequences, and this is something that will be on your quiz, so be sure to pay attention to this portion here. So what that basically, well, we're going to go through it, but what this basically means is you can look at the genetic coding or the base pair nucleotides, and you can determine who potentially came first, and you'll be able to place them on a cladogram. So in this example here, we have 7 species, species A through G. So we have species A, B, C, D, E, F, and G. So we have all 7 of those, and they all have a specific genetic code. And I've listed them here above me. So you can see all of their sequences. They have 10 base pair nucleotides there. And basically we're going to have a mutation that changes the DNA sequence, okay? And it's going to be analogous to a change of a characteristic. So we're going to pretend that that change in that base pair sequence changes a specific characteristic in those individual organisms or species. So that mutation's occurring, it's changing something, we don't know, we're only looking at the genetic code in this case. So over here we have up above, you have the 10 base pairs you can see, and this is our evolutionary ancestor here. This is our common ancestor. So this is going to be what we're going to be starting with for our cladogram when we're talking about the nucleotides and the base pairs here. And then we're going to look at all 7 of our, right here, we're going to look at all 7 of our species, and we're going to try to place them all in a cladogram based on their nucleotide and their genetic code here. So the next slide, I've gone ahead and placed what the answers are up above me here, and this is figure 2610. But you can see here we have our ancestor with the 10 base pairs. So that's our ancestor, our post-primitive sequence. So that's what we're starting with. So that's our GG, TAT, AA, CCC. Now, looking at all of the code, you can see here that species E has the same genetic code as its primitive ancestor in the sequence. So we're going to probably put that first because it existed first and it didn't change from the proposed primitive ancestor. So we're going to put that at the very beginning of the cladogram, so it came around first. Now, you can see here the next change that we're going to see is our 6th base pair nucleotide, which is an A. That got changed to a T. So the first change you can see here is in species D. So instead of a GGTATA, you're going to see GGTATT. So we're going to put that next because there's only one base pair change in location 6, and it only changed from an A to a T. So the next one we're going to place is going to be our species G, because now we have 2 base pair nucleotide changes. So in addition to changing the 6th nucleotide pair, we're also changing the 5th one. So we're changing from a T to a G. So instead of having GGTATA, you're having GGTAGT. So we're changing 2 of those nucleotide base pairs. Now, the next segment here gets a little tricky because we're changing a few different ones. So it's branching out a little bit differently. So we are changing, for species F, 1 more nucleotide base pair. So that's going to come over here on this 4th little stem coming off. So our 10th nucleotide base pair changes from a C to an A, so we're going to mark that down for species F. But for A, B, and C, we have a few differences that didn't quite change exactly the same as our species F. So we can see here that they don't all end in A. They do all end in C's or G's up here, so they've changed somewhat, at least species C has, but they've changed in other respects as well. So for species A, you can see here that the 2nd base pair nucleotide changed from an from a G to an A. And so now you're putting species A there. And the next one, you have the 7th base pair nucleotide changing from an A to a T in species B, so we're putting that one there. And then also you can see that it's the same in species C. We do have the 7th base pair in species C that's changed to a T, but in addition to that, we also have the 10th base pair is changed to a G. So instead of having it over here like we had in species F, we had the 10th base pair changed to an A. Over here we had the 10th base pair changed to a G. So that resulted in the divergence here of those two species, species F and C, slightly differently. So this is probably the best proposed way that these species came into existence, and again, each of those nucleotide base pair changes might indicate maybe that they have fingers or thumbs, or they might have, ears that are slightly different, or a tail that's slightly different. So they're all indicating that there's a characteristic that has changed slightly. So there's really a few steps in constructing a cladogram, like that video that we just watched. And we'll go through the steps. I believe there's 5 of them, and so they're pretty simplistic, but this is the general approach you want to follow if you're asked to construct a cladogram, which you are asked to do in our lab that we just did on the communicules. So the first step here is to choose your species. So for example, in our lab, we're using the communicule species, but this could be anything. This could be, a dog species, cat species, bovine species, whatever species that we're talking about at that moment in time. So the first step is choose a species. The second step is choosing your characters. So if you're choosing a few different types of species, make sure you're choosing characters that indicate that they are different. So for example, if you're choosing birds, any types of birds, some of them have different types of beaks, or different types of perching foot, or birds of prey have different type of feet. They have more predatory, locking with sharp talons. So make sure when you're choosing your species, you're also choosing characteristics that set them apart. So you're choosing your characters. And the third step here is you're going to determine the polarity of the character states. So are they primitive or are they derived? Are they coming from a primitive ancestor, like the ancestral eye? All birds have the same 2 sets of eyes, and that is an ancestral trait, a very primitive trait coming from your primitive ancestor. Or they can have derived characteristics, for example, as different types of wings, or different types of talons, or whatever we're talking about, any sort of derived characteristic that is inclusive to a specific in-group and slightly different than what the out-group would be having. The fourth step here is to analyze our cladogram. So for all species, even in higher taxa here, we're placing them at the tips of the tree in our phylogenetic tree. So we're not placing them at branch points, we're placing them at the tips of the tree. And it's important to note that each cladogram branch point should have a list of 1 or more of those shared derived characters. And those are the ones that are common to all of our species above a branch point, unless the character is later modified, so unless it's changed later in time. And then also to keep in mind that all shared derived characters appear together only once in a cladogram, unless they arose independently during evolution more than once. So unless they're arising independently and not dependently of any sort, we're making sure that they're arising separate from one another if we have them more than once in our cladogram. Okay, step 5 here is choose the most likely cladogram among possible options. the point of least resistance or the pathway of least resistance is typically the one that's going to be correct. So you're going to pick the cladogram that makes the most sense. Just like that video stated with the glass of milk. You know, if he comes downstairs, finds a glass of milk, it's spilled, likely it's his cat, not aliens or Bigfoot or Sasquatch. So you want to choose the most plausible, the most probable option here, the one that's the easiest, that makes the most sense in most cases. But again, these are just guesses, so we're not quite sure. And then lastly here, the sixth step is to choose a non-controversial outgroup as a root. So one that doesn't differ from the norm exponentially here. If we're talking about an outgroup, if you're going to be mentioning an in-group, choose an outgroup that is non-controversial, like doesn't have any sort of really strange, wacky things happening with it. Okay, so you also heard the mention of parsimony. So basically what the principle of parsimony is that the preferred hypothesis for our cladograms is the one that's simplest, as I mentioned before. And this includes the simplest explanation for the characters and all their states. When we when we have challenges in a cladistic approach, there is a challenge, and that's determining the correct polarity of events. And this is not always obvious. So it's not obvious which traits are primitive. You know, you don't know which ones came earlier all the time, and which ones are derived or which ones came later in evolution. Sometimes folks mess that up. So you just really have to evaluate and choose the simplest and try to determine the correct polarity of events. And sometimes we do use fossils to help resolve this question, but it's not always helpful because you don't always know what sort of, it depends on what sort of characters you're talking about. Any sort of cartilage-based characteristic might not be evident in a fossil, so you're not always sure what you're discussing here. So let's talk about an example of parsimony. So we're going to use the base pair nucleotides again to show this parsimony example here. So in this case we have 4 taxa, or 4 groups of species here. And so this is species A through D, so species A, B, C, and D. We have our primitive ancestral species here. You can see him right above 1, 2, 3, 4, 5. He's GTACA, so we'll use that one as our primitive ancestral species here. And in this case, we're going to select species A as the outgroup because it has all of the primitive states. It has all of the same primitive states as our primitive ancestor here. So now we're going to develop 3 potential trees in placing the species B, C, and D, because we want to see which one is most likely to make the most sense. And the answer, the one that's going to be the most parsimonious, is tree 3 because it requires the fewest number of mutations here. And I'll show that in the next slide here. So remember, we're trying to place species B, C, and D appropriately in a cladogram using the ancestral primitive species as our base. Okay, so here's our 3 trees, if you can see those okay. So according to this principle of parsimony, or the path of least resistance, the easiest, the simplest, that makes the most sense, tree 3 over here on this side of me is the most likely choice because it only requires 5 mutations, okay? So for example, when we're looking at that primitive species at the very bottom as our ancestral species, we're not changing from GT to GA to GA, then back to GT. So you'll see that happening in tree 1 and 2. Whereas species A has GT, but then species B has GA, then it changes back to species C in GT for the nucleotides, and then back to GA in species D. So it gets all crazy. So the mutations are going back and forth and back and forth. So likely that's not what's happening. Same thing with tree 2 here. So you'll see in species A it has GT, then species D is GA, species B is GA, and species C is GT. So it's going GT, GA, GA, GT, back to GT. So that is likely not to make a lot of sense. So I'm only looking at the first 2 base pair nucleotides in this case because it's pretty obvious to me. Now the third tree, we're looking at the primitive ancestor here, GTACA, and it's changing slightly, but it's only changing 5 different times over the course of, if you're looking at all of the base pair nucleotides. But the first 2 base pairs, we're going GT, GT, GA, GA, which is likely we're not changing as many times. So this is going to be the best likely answer because there's the fewest number of mutations that's occurring through the evolutionary timeline that's occurring here. So that's a really good way to pick a parsimonious answer when you're looking at base pair nucleotides and creating a cladogram from that. The least number of mutations is likely the right answer when you're looking at how species have changed in that cladogram over time. Okay, so that is all I have for today's lecture session. And so we covered cladograms and cladistics today, and the next lecture that I post is going to cover the remaining 2 sections of our Module 9, and so that will be posted here shortly, and I appreciate you guys listening.